Alright, Chapter 13 looks at host defenses, specifically with the third line of defense, and that is specific immunity. Then, at the end of the chapter, we will finish up with the process of immunization. So here are the total list of objectives for Chapter 13. This is quite lengthy, objectives 1 through 19. This chapter is just filled with detail, filled with information. I believe I mentioned this with Chapter 12, but immunology, the study of the immune system, is its own course. It can be its own course, it's its own chapter, it's its own textbook. So your list of chapter objectives here from just one chapter in your textbook could actually cross over to 2, 3, maybe even 4 separate chapters in an immunology textbook. So even though it looks like a lot, in all reality, we really are just kind of touching the surface of this information. And so hopefully, while you will take away a lot of detail from this chapter, we are still really focusing on the big picture. All right, in section 1, we're going to look at why the third line and how the third line of defense is different from the first and second lines. We're going to look at different terms such as antigen, immunogen, and epitope, and be able to define those and understand those. And we'll also revisit those terms in section 3. We will look at the 4 stages of a specific immune response, and then also discuss the role of cell markers within this whole process of the immune response. So the third line of defense is specific. All right, so with specificity, that means you have to have an antigen or an immunogen in which the immune system can be specific for. So antigens are immunogens. Immunogens are just molecules that elicit an immune response. The antigen, same definition, just 2 different words to use it, are molecules that stimulate a response by B and T cells. And this is also one of the main differences between the first and second line of defense and the third line of defense, and that is the players. Within specific immunity, your players are the B cells and the T cells. So an antigen can be a protein, it can be a polysaccharide molecule, meaning more or less a sugar, and these are going to be molecules that are on either the inside or the outside of cells, part of viruses, anything that is part of the microorganism. And it can be any exposed protein that's on the surface. It can be a protein that is released from a cell. It's going to be a foreign molecule that can stimulate an immune response. All right, and so a foreign molecule, we may be focusing here on microorganisms, but how many people out there have food allergies? All right, they have a peanut allergy, they have an egg allergy, they have a medicine allergy, penicillin allergy. All right, any of those molecules, they are actually foreign to our body, and so our immune system can identify those as foreign and, being a negative object, elicit an immune response, and then you develop those allergies. So, with an antigen, all right, antigens are going to be these whole proteins, these whole polysaccharide molecules, and within a protein there can be different parts. All right, so if you remember that proteins are made up of individual amino acids being strung together through peptide bonds, a section of that strip of amino acids is its own epitope. So the epitope is just kind of like the fingerprint. You have a handprint. All right, if you were to paint your hand, you could, and then put it against a piece of paper, you have a handprint. But part of that handprint, you have fingerprints that can be used for identification purposes. So then, if you kind of think about that, Your handprint is the big picture. Your fingerprints are part of that handprint, but they're also their own entity. And so in this case, the antigen would be the entire handprint, while the epitopes would be the individual epitopes. Alright, so 2 features that characterize the third line defense and definitely differentiated from the first and second line defenses are going to be specificity and memory. So it is very specific in terms of the components of the third line of defense are going to be specific for one type of organism or one type of antigen. So antibodies, which are produced within the third line of defense, if antibodies are produced against chickenpox virus, they're not going to function against the measles virus and vice versa. Then there's the memory component. And with this, you can have lymphocytes that have been basically programmed to recall their first engagement with a different antigen and then rush to attack once more. So that is why it's good to have vaccinations, because that vaccination basically gives your specific immune system a taste of the infection, a taste of the microorganism. And then when you come into contact with it for real in the environment, all right, your immune system has already seen it. It's already responded against it to a small degree, but now it's ready to go quickly to respond a second time and do it quickly, and so hopefully then you won't actually get sick with whatever you came into contact with. Alright, so here is figure 13.1, and it is overview of the stages of lymphocyte development and function. And so what this slide actually shows you are the 4 stages of the specific immune response. Alright, so you want to list these out. The first stage is going to be lymphocyte development and differentiation. Alright, and that's going to be in this first section here, where you have the lymphocytes being the B cells and the T cells that are differentiating from a stem cell, and they are basically developing. The B cells develop in the spleen. You got the B cell line right here, and the T cell line is going to develop in the thymus. All right, the second stage is going to be the presentation of antigens, and you have your antigen right here. It's going to be picked up by antigen-presenting cells, or a dendritic cell in this case, these purple guys with those dendrites. And the dendritic cell is going to process the antigen and then present it to a B cell or a T cell. And so here you actually have that specific presentation of the antigen, all right, that little blue guy there, to the T cell. So that's the second stage. And then the third stage is the challenge of B cells and T cells. So this is going to be the second time around. And then you have the actual response. All right, so that response is going to be activating the T cell, all of the different types of T cells to respond against that initial antigen, and then the production of antibodies that are going to move through the vasculature, through the blood, all in case this little antigen comes back to you, all right, comes back to your body. And now the material down here, these T cells, these antibodies are going to be ready to respond to the antigen again and take it down very quickly. All right, so concept check. B cells mature in the blank and T cells mature in the blank. Both cell types then migrate to the blank and the blank. All right, so if we go back to this state or figure here, all right, B cells. They're going to develop in the bone marrow, and then they migrate out to the lymph node and the spleen. All right, the T cells develop in the thymus and migrate out to the lymph nodes and the spleen. So, the correct answer here would be the bone marrow and the thymus. B cells mature in the bone marrow. T cells mature in the thymus. Both cells then migrate to the lymph nodes and the spleen. And that is where we are going to stop section 1.